Assembly PCR. Assembly PCR is used to assemble two or more pieces of the DNA into a single piece of DNA. The principle behind the assembly PCR is the synthesis of long DNA structure by performing PCR on a pool of long oligonucleotides with short overlapping segments. So in long oligonucleotides, they overlap with short overlapping segments to assemble two or more pieces of DNA into a single piece of the DNA. Here we can see that how two parts of the DNA, they are assembled with each other to form a single full length product. Here we can see that there are two parts, part A and part B. Part A is amplified with left primer and with right primer, while part B is amplified with right primer and with left primer. Once they are amplified, then they are again amplified with the help of the left primer and the right primer in case of to form a full length product. Here we can see that this is the left primer and this is the right primer, and the product is amplified with the help of left and right primer to generate final full length product. So assembly PCR is such a PCR in which there is initial PCR with primers that have overlap, and then there is a second PCR using the product as the template that generates the final full length product. So we can say that the assembly PCR is such a PCR in which we assemble two or more pieces of DNA into a single full length product.